And now to increase the number of copies of an integrated heterologous gene and thereby increase the overall yield of the recombinant protein, the gene can be targeted to yeast, repetitive DNA sequences such as the sequences ribosomal DNA RNA, encoding ribosomal RNA that are present in approximately 200 copies. Alternatively, the heterologous gene can be integrated into several of the 400 copies of delta sequences in the SRE-VCA genome, delta. The delta sequences are parts of rare non-essential DNA elements derived from retrotransposons. Briefly, retrotransposons are chromosomal DNA sequences that are transcribed but not translated into protein. The RNA molecules act as templates for the synthesis of DNA by the enzyme reverse transcriptase, and the copied DNA sequences integrate into the chromosome at different sites. In one study, 10 copies of a heterologous gene were inserted into alpha sequences and produced a significant amount of the recombinant protein. A YAC is designed to clone a large segment of DNA, 100 kilobase pairs, which is then maintained as a separate chromosome in the host yeast cell. The YAC system is highly stable and has been used for physical mapping of human genomic DNA, the analysis of large transcription units, the formation of genomic libraries containing DNA from individual human chromosomes. A YAC vector mimics a chromosome because it has a sequence that acts as an origin of DNA replication, ARS, a yeast centromere sequence to ensure that after cell division, each daughter cell receives a copy of the YAC and telomere sequences that are present at both ends after linearization of the YAC DNA for stability. In some cases, the input DNA is cloned into a site that disrupts a yeast marker gene. In the absence of the product of the marker gene, a colorimetric response is observed when recipient cells are grown on a specialized medium. Alternatively, some YAC vectors contain a selectable marker gene that is independent of the cloning site. To date, YACs have not been used as expression systems for the commercial production of heterologous proteins, although they have the protein to produce large amounts of either a single protein from multiple copies of the same gene or a heterologous protein with different subunits. Intracellular production of heterologous proteins in SRE-VCA. Right? Now, most SRE-VCA intracellular expressions systems have the same basic features. Here, the production of the human enzyme superoxide dismutase, SAD, SOD, will be used to illustrate the process. Superoxide anion is its byproduct of oxygen utilization in aerobic organisms. In humans, this anion helps both to stimulate the inflammatory response of phagocytes and to direct leukocytes to the site of an infection. However, too much of the molecule and its derivatives can cause cellular damage. To minimize these potentially cytotoxic effects, the naturally occurring cytoplasmic enzyme copper zinc SOD scavenges the superoxide radical and combines it with a hydrogen ion to form hydrogen peroxide, which in turn is degraded to water and oxygen by catalase or peroxidase. Superoxide anion is also produced when blood is allowed to re-enter an organ, reperfusion, after it has been deprived of blood during a surgical procedure. To prevent the source of superoxide anion damage, clinicians have speculated that copper zinc SOD could be administered to an organ as it is being reperfused. In addition, copper zinc SOD might act as a therapeutic agent against inflammatory diseases and such as osteoarthritis, rheumatoid arthritis, scleroderma, and ankylosing spondylitis. And for both of these uses, an authentic human form of copper zinc SOD is preferred to avoid any adverse immunological responses that might result from using an enzyme from another species. Initially, a complementary DNA, cDNA for human copper zinc SOD was cloned into an E. coli expression system. As expected, the E. coli host cells removed the initiator N-terminal methionine from copper zinc SOD protein. However, the next amino acid, alanine, was not acetylated as it is in human cells. To produce a fully functional protein, the human copper zinc SOD cDNA was cloned into a YEP vector. This YEP vector contained 1. A 2 micro M plasmid origin of DNA replication, 2. A yeast gene for leucine biosynthesis, LU2 for selection in yeast, 3. An E. coli origin of replication, 4. The ampicillin resistance AMP gene for selection in E. coli, and 5. The human copper zinc SOD cDNA inserted between the promoter region of the yeast glyceraldehyde phosphate dehydrogenase gene, GAPDP, and a sequence containing the signals from transcription termination and polyadenylation of mRNA from the same gene. A leucine defective LU2 yeast strain was transformed with a vector, and cells were plated onto medium that lacked leucine. Only cells with a functional LEU2 gene, which was supplied by the vector and could grow under these conditions. The GAPD promoter is transcribed continuously, constitutively during cell growth. In this experiment, the yeast cell produced high levels of intracellular copper zinc SOD that, like the authentic protein from human cells, had an acetylated N-terminal alanine residue. Secretion of heterologous proteins by S-serine. All glycosylated proteins of S-serine are secreted, and each must have a leader sequence to pass through the secretory system. Consequently, the coding sequence for recombinant proteins that require either O-linked or N-linked sugars for biological activity must be equipped with a leader sequence. Right? And to pass through the secretory system. Consequently, the coding sequences of recombinant proteins that require either O-linked or N-linked sugars for biological activity must be equipped with a leader sequence. Usually, the leader sequence from the yeast mating type alpha factor gene prepared pre-pro alpha factor is inserted immediately in front, upstream, the cDNA of the gene of interest. Under these conditions, correct disulfide bond formation, proteolytic removal of the leader sequence, and appropriate post-translational modification often occur, and an active recombination protein is secreted during the process. The leader peptide is removed by an endoprotease that recognize the dipeptide as shown lysine and arginine or arginine. The LIS-R codons must be located adjacent to the cDNA sequence so that following removal of the leader peptide, the recombinant protein will have the correct amino acid residue. Now figure 7.7 shows this S-series expression vector. The cDNA for human copper zinc SOD was cloned between the promoter GAPDP and termination polyadenylation sequence of the glyceraldehyde phosphate dehydrogenase gene. The LU2 gene that was cloned between segments of the yeast 2 micrometer plasmid DNA encodes a functional enzyme of the leucine biosynthesis pathway. The yeast origin of a replication is included in the 2 micrometer plasmid DNA. The ampicillin resistance AMP gene and the E. coli origin of the replication RI are derived from plasmid PBR322. This is the diagram of that. Please take a note. Now we have a milestone to be read. Synthesis of rabbit beta-globin in cultured monkey kidney cells following infection with SV40 beta-globin recombinant genome. Right? Courtesy R.C. Milligan and B.H. Howard and P. Berg, 1979. Thank you, sirs. Conceptually, the development of a eukaryotic expression system appears to be a relatively simple matter of assembling the appropriate regulatory sequences, cloning them in the correct order into a vector, and then putting the gene of interest into this precise location that enables it to be expressed. In reality, the development of the first generation of eukaryotic expression vectors was a painstaking process following a trial and error approach. Before Milligan, Howard, and Burke's study, a number of genes had been cloned into the mammalian SV40 vectors, but mature functional mRNAs were never detected after infection of host cells. This problem was overcome by inserting the rabbit cDNA for beta-globin into an SV40 gene that had nearly all of its coding region deleted but retained all the region implicated in transcriptional initiation and termination, splicing, and polyadenylation. Both rabbit beta-globin mRNA and protein were synthesized in cells that were transfected with this beta-globin cDNA SV40 construct. Mulligan et al. Concluded the principal conceptualizing innovation is the decision to leave intact the regions of the vector implicated in the mRNA processing. The study established that an effective eukaryotic expression system could be created by placing the cloned gene under the control of transcription and translation regulatory sequences. It also stimulated additional research that pinpointed in detail the structural prerequisites for the next generation of eukaryotic expression vectors. Right, and on to the subject matter proper at its N-terminus, for example, a properly processed active form of the protein heridin was synthesized and secreted by a Cerevisiae strain containing a YEP vector. That had the pre-pro alpha factor sequence added to the heridin coding sequence. The gene for heridin is from an invertebrate, leech Hirido medicinalis. This protein is a powerful blood anticoagulant that is not immunogenic in humans. Over the past 10 years, the amount of heterologous protein that can be produced per liter of yeast culture has increased 100-fold from about 0.02 to 2 grams per liter. This increase is mainly due to the improvements in growing cultured cells to high cell densities, and the level of protein produced per cell has remained largely unchanged, although there have been significant advances in techniques to increase the number of copies of a target gene in a host cell and to increase the expression levels of these genes. The overexpressed proteins tend to form intracellular aggregates, often associated with molecular chaperones, rather than to be secreted into the medium, which facilitates purification. Major problems that must be addressed to increase the heterologous protein secretion in yeast cells are the incorrect folding of the polypeptide, the activation of cellular mechanisms to cope with the stress of protein overproduction, and the aberrant processing and release of the protein of interest from the endoplasmic reticulum. One of the major reasons for producing a recombinant protein for use in human therapeutics in yeast rather than in bacteria is to ensure that the protein is processed correctly following synthesis. Correct protein folding occurs in the endoplasmic reticulum in eukaryotes and is facilitated by a number of different proteins, including molecular chaperones, enzymes for disulfide bond formation, signal transduction, proteins that monitor the demand and capacity of the protein folding machinery, and proteases that clear away improperly folded or aggregated proteins. The eukaryotic enzyme protein disulfide isomerase is instrumental in forming correct disulfide bonds correct disulfide bonds within a protein. Aberrant disulfide bond formation changes the protein's configuration, which abolishes protein activity and causes instability, poor yields of overexpressed proteins that occur because. Take a screenshot of this and we'll come back to the details later. So the capacity of the cell to properly fold and secrete proteins has been exceeded. Several strategies have therefore been implemented to increase the host cells capacity to process higher than normal levels of proteins. For example, the overproduction of molecular chaperones and protein disulfide isomerases might increase the yield of recombinant proteins, especially those with disulfide bonds. To test this hypothesis, the yeast protein disulfide isomerase gene was cloned between the constitutive glyceraldehyde phosphate, dihydrogenase promoter, and a transcription terminator sequence in the YRP vector, and the anti-construct was integrated into a chromosomal site. The modified strain showed a 16-fold increase in protein disulfide isomerase production compared with the wild-type strain. When protein disulfide isomerase overproducing cells were transformed with a YEP vector carrying the gene for human platelet-derived growth factor B. There was a 10-fold increase in the secretion of recombinant protein over that of transformed cells with normal levels of protein disulfide isomerase. The overproduction of protein disulfide isomerase specifically increases the secretion of proteins with disulfide bonds. Higher levels of secreted products are also obtained for the recombinant proteins human erythropoietin, bovine prochymosin, and leech hirudin, and SRVC cells that overexpress the chaperone BIP. Overexpression of molecules chaperone BIP or protein disulfide isomerase increase the secretion of some heterologous protein. However, overexpression of a single chaperone may not have the desired outcome. Onto the diagram that we just saw. Summary of protein folding in the endoplasmic reticulum of yeast cells during synthesis on ribosomes. Associated with the endoplasmic reticulum, nascent proteins are bound by the chaperones VIP and calnexin, which aid in the correct folding of the protein. Protein disulfide isomerase PDI catalyze the formation of disulfide bonds between cytosine amino acids that are nearby in the folded protein. Quality control systems ensure that only correctly folded proteins are released from the ER. Proteins released from the ER are transported to the Golgi apparatus for further processing. Prolonged binding of VIP to misfolded proteins leads to activation of the SRVC transcription factor HAC1, which controls the expression of several proteins that mediate the unfolded protein response. And in some instances may increase the degradation of the target protein. This is because proper protein folding requires the coordinated efforts of many interacting factors. Even when levels of even chaperone are adequate, the levels of co-chaperones or cofactors may be limiting. The unfolding protein response of each cell coordinates the expression of several chaperones as well as co-chaperones. When the demand for protein folding exceeds the folding capacity of the endoplasmic reticulum, the unfolded protein response increases the expression of chaperones, protein disulfide isomerase, and other proteins involved in protein secretion. Engineering the proteins with unfolded protein response may be a better approach to increase the overall capacity of the cell to fold proteins in a coordinated way by maintaining appropriate rations of all factors required. Accumulation of unfolded proteins in the endoplasmic reticulum activates the SRVCA transcription factor HAC1, which activates the expression of proteins of the unfolded protein response, and therefore expression of HAC1 was targeted for improved secretion of the important industrial enzyme alpha amylase, which is used for starch hydrolysis in a wide range of processes such as alcohol production, paper recycling, and oil drilling. The peculiar Pasteur's expression systems. Recombinant proteins have been produced successfully in cerebecia from cloned genes from many sources. However, in many cases, expression levels are low and protein yields are modest. One of the major drawbacks of using cerebecia is the tendency for the yeast to hyperglycosylate heterologous proteins by typically adding 50 to 150 mannose residues in end-linked oligosaccharides side chains that often alter protein function. Although the initial stages of addition of glycan chains to proteins in the lumen of the endoplasmic reticulum are similar in yeast and humans, following transference of the protein to the Golgi apparatus, further processing differs significantly. The outcome is the production of a sialylated protein in humans and a hypermanosylated protein in yeast, with alpha-1,3 bonds between the sugar residues that can make the heterologous protein antigenic. All the proteins that are designated for secretion frequently are retained in the periplasmic space, increasing the time and cost of purification. Finally, cerebecia produces ethanol at high cell densities, which is toxic to the cells, the Crabtree effect, and as a consequence lowers the quantity of secreted proteins. For these reasons, researchers have examined other yeast species and eukaryotic cells that could act as effective host cells for recombinant protein production. P. pasteuris is a methylotrophic yeast that is able to utilize methanol as a source energy and carbon. It is an attractive host for recombinant protein production because glycosylation occurs to a lesser extent and the linkages between sugar residues are the alpha-1,2 type, which are not allergic to humans. With these natural characteristics as a starting point, a pasteuris strain was extensively engineered with the aim of developing a humanized strain that glycosylates proteins in a manner identical to that of human cells. Both human and yeast cells had the same small 10 residue branched oligosaccharide to nascent proteins in the endoplasmic reticulum. However, this is the last common precursor between the two cell types because once a protein is transported into the Golgi apparatus, further processing is different. In the Golgi apparatus yeast cells, they add an alpha-1,6 mannose residue to the oligosaccharide, which subsequently leads to hypermanosylation. Mammalian cells, on the other hand, remove some mannose residues from the precursor, trimming and then subsequently add specific sugars to yield a glycoprotein with an oligosaccharide that terminates in sialic acid. To create a humanized strain, the enzyme responsible for addition of the alpha-1,6 mannose was first eliminated from P. pasteuris to prevent hypermanosylation. Next, the gene encoding a mannose trimming enzyme, a manosidase from the filamentous fungus, Trichoderma ricei, was inserted into the yeast to genome and was found to trim the oligosaccharide to a human-like precursor. Genes encoding enzymes for the sequential addition of sugar residues that terminate the oligosaccharide chains and galactose were also added. It should be noted that the coding sequence for all engineered genes contain a secretion signal for localization of the encoded protein to the Golgi apparatus. Finally, several genes for proteins that catalyze the synthesis transport to the Golgi apparatus, in addition of sialic acid to the terminal galactose on the protein. The precursors were inserted into the P. pasteuris genome. Properly sialylated recombinant proteins that can be used as human therapeutic agents have been produced by the humanized P. pasteuris, including erythropoietin antibodies. During growth on methanol enzymes required for catabolism of this substrate are expressed at very high levels with alcohol oxidase. The first enzyme in the methanol utilization pathway encoded by the gene AOX1, representing as much as 30% of the cellular protein. Transcription of AOX1 is tightly regulated. In the absence of methanol, the AOX1 gene is completely turned off but responds rapidly to the addition of methanol to this medium. Therefore, the AOX1 promoter is an excellent candidate for producing large amounts of recombinant protein under controlled conditions. Moreover, the induction of the cloned gene can be trimmed to maximize recombinant protein production during large-scale fermentations. In contrast to SCR-ABC, P. pasteuris does not synthesize ethanol, which can limit cell yields. Therefore, very high cell densities of P. pasteuris are attained with the concomitant secretion of large secretions of protein. P. pasteuris secretes very few proteins, thus simplifying the purification of secreted recombinant proteins. Many of pasteuris expression have been devised, each one having more or less the same format. The basic features include a gene of interest under the control of promoter and transcription termination sequences. For the P. pasteuris AOX1 gene, an E. coli origin of replication and selectable marker gene, and a yeast selectable marker gene, figure of 7.1. The addition of a signal sequence from either the pasteuris phosphatase PHO1 gene or another yeast gene facilitates the secretion of a recombinant protein. To avoid the problems of plasmid instability during long-term growth, most P. pasteuris vectors are designed to be integrated into the host genome, usually within the AOX1 gene, the HS4 gene for histidine biosynthesis, or rDNA. Both the engineered gene of interest and its yeast selectable marker gene are inserted together into a specific chromosome site by either a single figure 7.11a or a double 7.11b recombination event. The P. pasteuris logically active P. pasteuris expression system has been used to produce more than 100 different biologically active proteins for bacteria, fungi, invertebrates, plants, mammals, including humans. Many of these proteins, such as the hepatitis B virus surface antigen, human serum albumin, and bovine lysozyme, are identical to the native proteins. The illustration shows P. pasteuris integrating expression vector, the gene of interest. GOI is cloned between the promoter AOX1, P, and termination polyadenylation. Sequence AOX1T of the P. pasteuris alcohol oxidase 1 gene, the HIS4 gene encodes a functional histidinal dehydrogenase of the histidine biosynthesis pathway, the ampicillin-resistant ZAMP gene, and an origin of replication. Function of E. coli, the segment marked 3, AOX1 is a piece of DNA from the third and 3' end of the alcohol oxidase 1 gene of P. pasteuris, a double DNA recombination event between the AOX1P and 3' AOX1 region of the vector and the homologous segments of chromosomes. Other yeast systems, authentic heterologous proteins for industrial and pharmaceutical use, have also been generated in other yeasts. For example, cDNAs for the alpha and beta-globin A were each cloned between the methanol oxidase promoter MOXP and transcription terminator MOXT sequences of the methylotrophic yeast, hence cellular polymorpha, and placed in tandem in expression vector. Fortuitous integration into a chromosome yielded an isolate that produced functional hemoglobin, that had the correct tetrameric organization of the 2 alpha and 2 beta, globin chains, alpha to beta to alter large amounts of the animal feed enzyme supplement phytase have been produced by transformed H polymorpha. The thermotolerant dimorphic yeasts, Oxula, adenovirinates, Gerovia, diplotica, labellitica have demonstrated promising potential as hosts for high levels of heterologous proteins expression. The yeast can grow at temperatures at 48 degrees centigrade and can survive at high temperatures 55 for several hours. At high temperatures, the fungi grow in a mycelial form and revert to budding cells below 42. Some secretory proteins such as glycosamylase and invertase produce at higher levels of mycelia. Cell morphology also increase. Cell morphology also influence post-translational modification with Olin glycosylation predominating in budding cells, or N glycosylation occurs in both mycelia and budding cells. And while N glycosylation occurs in both mycelia and budding cells, an additional advantage of adenovirus is the ability to grow on a wide range of inexpensive carbon and nitrogen sources. Stable chromosomal integration systems have been developed for adenovirus, including a promising system based on complementing of a tryptophan autograph, auxotroph. An expression cassette was constructed with the target alpha amylase gene MEA from the bacterium Bacillus amyloliquefaciens flanked by the AIIMS&NEET TF1 promoter, the S-CyRVC-PHO5 terminator, and the selectable marker ATRP1 that restores tryptophan biosynthesis in an AIIMS tryptophan oxytrophic strain carrying a deletion in the chromosomal copy of ATRP1. The 25S rDNA sequence was included on the vector and the expression cassette for targeted integration into the yeast chromosome following transformation of adenovirus with the vector and selection of a medium that did not contain tryptophan transformants were isolated, and synthesized tryptophan produced high levels of alpha amylase and carried a single copy of the vector with expression cassette within a chromosomal 25S rDNA site. To increase the number of copies of the expression cassette integrated into the chromosome, its effector promoter was used to drive expression of the ATRP1 selectable marker. The integration of multiple copies of the cassette compensated the low levels of ATRP1 expressed from this promoter and resulted in a sufficient synthesis of tryptophan to enable growth on a medium lacking tryptophan. Integration of 8 copies of MEA and up to 5-fold higher levels of alpha amylase activity. Moreover, by inserting the ATRP1 and MEA genes in the middle of the 25S rDNA sequence on the vector, following a double recombination event, only the expression cassette was integrated into the host common chromosome. Sequences present on the vector that are required for initial manipulation in E. coli were excluded. Going back to the diagram, a wide-range yeast vector system for expression of heterologous genes in several different yeast hosts. The basic vector contains a multiple cloning site, MSCS, for insertion of selected modules containing appropriate sequences for chromosomal integration, rDNA module, replication ARS module, selection module, and expression module for target gene in a variety of yeast host cells. Show samples of interchangeable modules, sequences for maintenance, and selection amp for the vector E. coli are also included. This lecture ends in 2 minutes time, and that will be a session for today, and the rest of it will be in the next session. So we have about 4 to 5 hours of lecture each day, right? And make notes on these, right? Slowly we sum up, and let me just sum up of any check out any diagrams left to show you. This is of the widespread range yeast vector system. This is the constructs of a stable integration of target genes into a chromosome of the yeast adenovirus, adeninovirus, target gene 2320. And this is the integration of DNA into specific B asterisk chromosome site 1 or double and B composite recombination and single recombination dashed line. The HIS4 gene of an intact circular plasmid and chromosomes HIS4 MUT results in the integration of the entire vector, including the gene of interest, with the AOX1 promoter and the F prime AOX1. Right, so that's it. We sum up now. The rest of it will be coming up in the next lecture. Please take screenshots of all these diagrams. They should be of help to you, right? Thank you, and I'll see you at the next lecture tomorrow. Best wishes. Make screenshots. Make sure you put your queries on the comment section, and I'll get back to you.